Yes, good morning, everybody. My name is Alexei Grom. I'm a pediatric rheumatologist in Cincinnati. And over the next couple of hours, we would like to discuss some of the challenges in drug development for patients with refractory systemic juvenile idiopathic arthritis and macrophage activation syndrome. And I would like to start with an example of a traditional successful design that actually was used in systemic GRA, systemic juvenile idiopathic arthritis in general, and explain why this particular design may not be suitable for this particular group of patients. So this was a clinical, actually two clinical trials of canakinumab in systemic juvenile idiopathic arthritis. There were two components. The eligible patients included those with active disease, and they all were required to have a stable treatment prior to the enrollment. Methotrexate was allowed as a background therapy, but it was supposed to be stable. The patient was supposed to be receiving a stable dose for at least 2 months. Biologics were not allowed. So already here we have a problem. If you think about macrophage activation syndrome in particular, the clinical picture there evolves very rapidly. And we typically start adding corticosteroids, we start adding cyclosporin. So it's very difficult to have a sort of a stable background treatment in this patient population. So again, going back to this trial, in the first part of the trial, all eligible patients were immediately randomized to receive either canakinumab or placebo in a blind manner. Again, giving a placebo to a very unstable child with in the ICU who may require escalation of the treatment is very risky and in fact not ethical. So that was the first part of the trial, and the effectiveness of the drug was assessed at day 15. And again, in this example, we are comparing responses in patients treated with canakinumab versus placebo. The second trial was much more complex. All the patients who received canakinumab and responded to canakinumab in trial 1 were included in this, enrolled in this particular trial, plus some new additional patients were enrolled as well. All these patients enrolled at this stage were started on the real drug. They all were receiving canakinumab for several weeks. And then those patients who showed a sustained clinical response to the treatment were, again, in a blind manner, randomized to receive either placebo or to continue receiving the drug. And what the investigators were looking for, they were looking for the differences in the number of flares between patients receiving canakinumab or placebo. And again, coming back to our patient population that we're talking about here, MAS would be very unstable, and an early withdrawal of a critical drug can actually provoke another life-threatening flare, and it could be, again, dangerous and, in fact, in many cases, unethical. So that's the reason we would like to have this session today and to hear the perspective of the parents, of the clinical investigators running clinical trials, as well as the representative of from the FDA and pharma, to see sort of their perspective on how these issues could be addressed. And I think we will start with the patients, with their perspective. Thank you.
I'll help kind of talk to the patient, and then I'll hand it back to Alexei for the panel itself. And Tara, can you please highlight all the panelists who are here, and the patients will be speaking, but we want to start seeing them if possible.
Yes.
Okay, and Chelsea, can we start with you?
Hey, sure.
Yeah.
Hold on just a second.
So Chelsea is the parent of a child with SJIA, and she has recently been in a clinical trial. And the other person we have here is Regina, and she has participated in a MAS drug trial, and Chelsea in an SJIA drug. And we'll hear a short experience from both of them. And I'll ask a question or two, and we'll try to have 1 or 2 questions for that you can, all of the panelists can ask as well.
Okay. Do you want me to start, Rashmi?
Yes, please do.
All right. Good morning. Like Rashmi said, my name is Chelsea O'Donnell. I have a 6-year-old with systemic JIA. She was diagnosed when she was 2, so we have been diagnosed for 4 years now. She has unfortunately never been controlled, so she's refractory. Her refractory is more arthritis than systemic features. She does have the rash and fevers, but her labs have always been quote unquote good. She's never been into MAS. So the way we got into clinical trial is that we had exhausted basically all of our options. We had stacked meds, we had pushed meds together, and she still was having this persistent joint damaging arthritis, and it was not going away despite joint injections, despite the medication. Nothing made it go away. It would go away in one joint, pop up in another. It was just very, very persistent. And so we tried Alaris first. And she was on tocilizumab plus leflunomide at the beginning of her trial. And those were pushed together. She actually started to develop an allergic response to tocilizumab. So we knew we had to come up with a different solution for her. And that was when our doctor suggested clinical trials. So what we did is we looked at 3 separate ones. We chose what we thought was the best fit for our family, which was a JAK inhibitor trial. And that's how we ended up in trial. We had to wait 10 months after we picked the trial that we wanted to use for her cohort to even open. And then after that 10-month period was over, and we entered into a 12-week washout. So not 12 days, but 12-week washout. So she could not have any biologic, any DMARD, anything like that. So that was very concerning because she obviously had this very persistent disease. So we knew she was going to get worse before she was going to get better. It was obviously a concern for, could she develop MAS if we pulled her off of these drugs? That was something that we had to weigh. And in fact, what happened is it was very coincidental timing, but the washout period began during COVID quarantine. So anything like going to school, and she was in pre-K at the time, going to school and germs and all of that stuff were kind of eliminated, which was good. And then we entered the summer, which is obviously non-URI season. So she did very well in the washout. Now her joint count did go up. The rashes did come back, but what was interesting is that her fevers, which used to be 104, 105, those did not come back whatsoever with the washout period. Though they stayed, the highest clocked was about 100.8. And our doctor told us that some systemic patients like our daughter that has not had MAS, a lot of times that research has shown that will do that the fevers and the other stuff will kind of simmer out and then just be the really persistent arthritis. So when we began clinical trial, her joint count was at 18, including her TMJ joint and hip joint, which, as all of you know, are the most kind of worrisome. That's about it. The only other thing that I wanted to say was systemically she did so well when we pulled her off of the drugs. Her labs were so good that we decided to give her the MMRV vaccine, which is very anxiety provoking for any parent, any SJIA parent. So we were afraid that could go into MAS or, make her very sick. But to her betterment, it did not. So that is our story with the washout.
Yeah. If I remember correctly, Chelsea, you told me that you had a specific discussion with your physician about the fear of MAS, and you kind of looked at her labs in the past, and she didn't have any of the MAS markers, correct? Can you just comment on that? So the risk was okay to take.
Yes. So in my daughter's case, we felt that the risk outweighed the benefit, and that is because she has never had macrophage activation syndrome, and two, she does not have the biomarkers that make it likely for her to develop macrophage activation syndrome. So with that was more comforting than a patient who, if we had experienced MAS or had the likelihood of developing MAS, obviously we couldn't have done a 12-week washout period.
Yeah. And I just want to comment on this for a minute that, I talked to a lot of patients, and it's really remarkable that all the refractory patients who have a history of MAS have never been in trials. So if the trials designs are not friendly towards patients like my kid and many of the other patients here, that means the drugs are being trialed on patients who are not like our kids. And there is something different about our kids who have a tendency towards MAS. So you end up with drugs which haven't been tried in this set of actually very refractory patients and with systemic patients. So this is something really worth keeping in mind as we think about trial design. Any quick questions for Chelsea from the panel, from anyone else? No. Okay. So we'll move on to Regina. Regina participated in a MAS trial last year. Yes, last year. Tell us about it, Regina.
Seems like it was just yesterday, but then an eternity ago was the way 2020 was. But good morning, everyone. You saw Juliana a few minutes ago. I'm her mom. Just a brief summary of her disease course. Juliana was diagnosed in July 2017 with a very similar onset of rash and fever, but she never had any active arthritis in the joints. She was put on Kineret, and she seemed to be doing well, but she always hovered in subclinical or smoldering MAS, which means she was clinically behaving okay, but her labs were never really normal. And then over the course of the last year, 2018, we started to notice reddening in her fingertips and toes. And it took about a year, but those symptoms kind of led me to push for CTs, and we found that she has interstitial lung disease. That was confirmed in May 2019. She was not doing well at the time, and her labs were trending high. Her ferritin was at about 1800 in May 2019. We had just tapered off steroids, so I was very hopeful. And at that point, her doctor said maybe we should try some pulse steroids just to kind of tamp down any smoldering labs. And MAS reared its ugly head very quickly. Within 2 weeks, her ferritin went from 1800. We upped her cyclosporin dose a little bit, and it went down to 1500. And then she had a fever. I brought her into the ER, and she was tested. Her ferritin was 30,000, and she was admitted right away. This was our first bout with fulminant MAS. She had always had these smoldering episodes where we really just changed medications to tweak and get her back on course. But my daughter was gravely ill. We were fighting with all traditional therapies. We had IV cyclosporin, IV prednisone. We upped her Kineret. She was on a regimen of 40 milligrams a day. We were up to 400 milligrams a day and still not getting control. And I just reached out to the SJIA Parent Network on Facebook, and I said what was going on. And the foundation reached out to me, and I learned about the clinical trial for Gamifant or emapalumab. I live in New York, and we learned that that clinical trial was only happening in Cincinnati, which is 12 hours or about 700 miles away. I was 8.5 months pregnant with my second child, and my husband husband and I had some really serious decision making because my daughter was declining rapidly. I don't think we had the luxury to wait. And overnight, her lungs started taking on fluid. So learning about the trial was kind of a last resort, and I'm so glad we did find out about it because she had a very overwhelmingly positive response to the trial. We were medically airlifted by Airbus from New York to Cincinnati and arrived. And within hours of her receiving imipalumab, she started eating, drinking. It was not an easy trial. The drug itself was an easy trial, but the commitment to it, we lived in Cincinnati for 5 weeks. I had to abandon my OBGYN, but at that moment, nothing is more important than saving your child's life, and I would have done anything. And I'm so grateful to the team there and the people that manufactured imipalumab because she's still here with us today. I will just say that it's a taste of something really good, and then you can't continue with the drug after the trial is over. My daughter had a great response, better than any biologic she had ever been on. And as that drug started to leave her system, her refractory disease returned, and we are still in a place where we're figuring out if we're stacking or switching. So my daughter's failed Kineret. She's failed Anakinra, and now we are trying Alaris and moving towards JAK inhibitors. So that's a little bit of where we are today, but the clinical trial was the best thing that came out of 2019 for our family.
Let me ask you, Regina, looking back, thinking about the way the trial was structured, which was maybe not top of your mind at the time, but suppose you had been told that you could participate in the trial, but you didn't know if you would get a placebo or the drug. Would you have made the trip to Cincinnati for that?
Really great question. I think in a moment like that, you are not taking no for an answer as far as there's no more treatments available. But to be honest, I talked about it with my husband. We make these decisions jointly, even though he's not here right now. But we would be very hesitant to participate, especially when it comes to something as fatal as MAS. We were never put in a situation like that. We did not have the time or luxury to think about stopping medications or washout periods or placebos. And that was a question that I asked in the beginning, and I was reassured there was no placebo arm of the study. And that was one of the major reasons we decided to move halfway across the country to take this medicine.
Yeah. And you mentioned washout. So, that would have been, what would your reaction have been if you had to stop? I mean, your daughter, like my child, we have similar situations, has SJIA, MAS, and the lung component. Yeah, there's 3 things going on. So suppose for the trial for the MAS drug, you had to stop the SJIA drugs. What would your reaction have been?
Well, I echo what Dr. Grom said earlier in that I think it would have been really risky. And, I think I'm going to say something that people wouldn't want to say. I don't think she would have survived. I don't think we had days to wait and play around, let alone weeks for a washout period. So, going back to what you said of these refractory patients, to pull them off drugs that they need to survive or to have some sort of quality of life to try. For me, it was a success story for Juliana. And I know that there was exclusion criteria to participate in the study, and we were just crossing our fingers that none of the medications she had been on would put us into that exclusion criteria. I do know that with Juliana as well, there was the desire per the trial to not have her on secondary agents like IV cyclosporin, but when she arrived at the doorstep of the trial, she was in horrible shape. There was no way they could pull her off certain medications for a washout. She needed a blood transfusion just to be stable enough to receive imipalumab. So no, I don't think we would have participated. I think we would have continued with IV cyclosporin and steroids, just knowing that she was getting something.
Okay. Are there any questions for Regina? Otherwise, we'll move on to the panel portion.
So I'm Randy Cron. I'm a pediatric rheumatologist in Birmingham, Alabama. And I'm going to briefly talk about a trial for the IL-1 receptor antagonist Anakinra to treat macrophage activation syndrome, kind of for all comers, including children and adults with Still disease. And the reason we undertook this is that for a while, I guess about 15, 16 years now, we've been using this particular drug to treat many types of macrophage activation syndrome, including our patients with Still disease. And for the bulk of patients, it works pretty well. But you ultimately need to do the clinical trial in a randomized, blinded fashion to prove to the greater community that this is beneficial. And I had a hard time convincing people to fund this for a long, long time. And eventually we got funding, but probably not enough to do the proper trial. So we kind of did a phase 2, 3-ish trial on a much smaller scale locally here in Birmingham. I do have disclosures. And I partnered with my adult rheumatology colleague, Wynn Chatham. I'm not a clinical trialist, although with COVID, I'm quickly kind of becoming one. Anyway, Dr. Chatham has a lot of experience doing clinical trials and is a great adult rheumatologist who treats Still disease and lupus and a variety of other things. And so we partnered to do both children and adults with macrophage activation syndrome. We were hoping to enroll 40 participants based on the money that we could get to perform this trial. And it is randomized, it is placebo-controlled, it is double-blinded to the patient and the investigators. And this was started about 4 years ago. And because we really wanted to prove benefit but not do harm, it's only really, although we collect data for about a month, the trial itself is only 3 days. And so once you meet entry criteria for the trial, you get randomized to receive placebo plus methylprednisolone at 500 milligrams, so a pretty substantial dose because we didn't want people to die and steroids work. And we wanted to compare that to the same dose of glucocorticoids plus Anakinra. And for the adults, or essentially anyone over 40 kilograms, that would translate to 100 milligrams every 6 hours, just for 3 days. And after the 3 days, it's up to the physician, who doesn't know if they're on Anakinra or not, to do whatever they feel is best, including Anakinra. And so that's the basic trial design. The primary outcomes were seeing if there were deaths. Unfortunately, that's kind of the hard outcome, or secondary infections during the process. And then the secondary outcomes were kind of measures of disease activity, primarily based on laboratory values and or whether they needed increased glucocorticoids, for example. And so we didn't want to include children with familial or genetic HLH or MAS, and so we excluded children under 1 year of age, but it was essentially open to everyone else. And this, I think we probably didn't do properly, but we tried to do the best up front. And that was if you had a diagnosis of systemic JIA, because we figured those children with Still disease would likely be a significant number of our populace who potentially would be enrolled in this, then you met the criteria that Angelo Ravelli and colleagues developed in 2016, because this is when this trial was done. I know there's recent criteria since then. Or if you didn't have a diagnosis of systemic JIA, we kind of mixed and matched things. And this is where I think it was pretty tricky, because we wanted to make sure that they had a cytokine storm and were sick. On the other hand, when you start looking at the exclusion criteria, we really wanted to make sure that people weren't horrifically sick. So first of all, we excluded patients with malignancy. Those patients are most difficult to treat in general. If they had a bacterial infection, something growing in their blood, we excluded them. And EBV is also a very difficult aspect of cytokine storms to treat, or MAS. And so we excluded if they had active EBV. And if they were on previous treatment for the current MAS episode, we had to exclude them as well. But I think where we really were careful was in patients who didn't have renal failure, which tends to be a later part of macrophage activation syndrome. When all your organs starting to fail, then your kidneys start to fail at some point too, and mechanical ventilation. So these were sick enough to be hospitalized, but not sick enough to be at death's door per se, or requiring pressor support was another one we excluded. So it starts dwindling down your potential patients pretty quickly. And so today we've actually only enrolled 8, and we probably will not pursue this further. I think we'll probably analyze the data, maybe include a couple more. And the hard part for us was, once again, it was just a single site study. And because both Dr. Chadman and I are rheumatologists, we essentially only enrolled mostly rheumatologic patients. So we had 3 adult onset Still disease, 3 lupus, one of which was a pediatric patient, and 1 JIA patient was not a Still disease patient, and 1 of kind of unknown trigger and etiology. So it's been challenging. I mean, and like I said, I'm not a clinical trialist. I'm quickly becoming one, in part because of COVID and trials we're doing there. But a lot of it does come down to trial design. And but I think ultimately we really want to get out the point that this is a safe, relatively safe therapy and hopefully effective. But that was at least our goal.
So Randy, you are still expecting that the data that you managed to generate in this trial would be convincing enough, or at this stage, you're not sure?
My goal is that we'll publish it and all the patients lived, whether they got placebo or not, but that placebo included high-dose glucocorticoids. And then after day 3, they could get whatever they want, which was typically Anakinra and steroids. So mostly we want to say that it didn't harm people. And if we can show benefit, and it'll likely be secondary outcomes, it's not going to be death for sure, that it's at least had some type of benefit. Because ultimately this can't, I don't think this could be done at a single site. Even though my personal belief is there's a lot of cytokine storm syndrome out there, depending on how you define your inclusion and exclusion criteria, this needs to be done on a larger scale. And so that's our ultimate goal, is just to kind of get this out there. And I think, you know, in the meantime, despite not having a randomized trial, except now with COVID, people are starting to use this more and more.
Randy, again, the way you address the issue here, you limited this placebo phase to just 3 days. And again, sort of after what you have done, what's your feeling? Is it sufficient? Is it?
Yeah, well, we haven't broken the code. We haven't analyzed the data, so I can't really say. Mm-hmm. But really, we wanted to be safe, and we wanted to make sure the patients got something, right? We were really terrified of not giving them glucocorticoids, for example. So.
All right, thank you. And I don't think there are any other questions to you. So I think we can move to Bas. Bas, I think you're next. Thank you, Randy.
Thanks, Alexei. I'm going to share screen as well. Do you see my screen now?
Yes.
So I run a clinical trial in the Netherlands in 5 of the 6 national academic centers. It's called the Early Stop of Targeted Treatment in Systemic JIA, or ESTIS trial. And this trial is really on the benign end of systemic JIA. These are my disclosures, and I have to acknowledge that besides a grant from the National Health Sciences Program, this grant is also sponsored in part by Sobi, although via a non-restricted research grant. They have no influence on the setup and on the results. And our trial acknowledged the observation that there probably is a window of opportunity early in the phase of systemic JIA, in which patients are at least higher responsive to IL-1 targeted therapy. So we treat our patients since 2008 with Anakinra as first-line treatment, so without the use of steroids. And we did it first in a prospective cohort study, and this was published in 2014 and 2019. So this is the flowchart of the prospective cohort study, not the trial. But in these patients, we started as a first-line treatment with Anakinra, 2 milligrams per kilogram per day. If there was persistent fever at day 3, we continued, but increased the dose of Anakinra to 4 milligrams per milligram per day, per kg per day. If there was persistent disease activity, at day 7, we either switched to another biological therapy, for example, anti-IL-6, or we added low to medium dose steroids. And the trial is really in this arm. So in those children that got into clinically inactive disease on Anakinra only, so without added steroids or switch to another biologic, we tried to taper and stop Anakinra over the course of several months in the first years of our study, and we found out that approximately half of those patients had to restart Anakinra pretty early again, but they clinically were in inactive disease, like those children that were able to stop. And the only difference that we could measure was at that time, at the time of tapering, actually a difference in IL-18 measures. So these are the results of the cohort study. I won't go into that. So this is the trial. It runs now in 5 out of 6 academic hospitals in the Netherlands. We aim to include 40 patients in the intervention arm of the trial. You've seen the outline, the flow of the study, and we tapered at in an interval of 6 months, from 3 months after start of therapy, if patients were in clinically inactive disease and if they had an IL-18 level of below 1200 picograms per liter, and if they had clinically inactive disease but had higher levels of IL-18, we kept them on Anakinra, and we measured and saw them again in an outpatient clinic 1 month later. So this is, I'm afraid it's partly in Dutch, but I think you can appreciate that there's an open label lead-in phase in which we just treat all our patients. We start with Anakinra. If those patients that achieve clinically inactive disease at month 3, they run into the intervention phase of the study. Every month their IL-18 level is measured. If it's below 1200, then they taper and stop. If it's more than 1200, they continue Anakinra. At month 9, we taper and stop anyways in any clinically inactive disease, even in those patients that have higher levels of IL-18. For now, we have included 46 patients in the open label lead-in phase and 32 patients in the tapering phase and in the intervention phase. So I estimate that we need 1 more year of inclusion to finalize this trial. But again, this is a trial really aimed at those patients that respond very quickly and very good to Anakinra monotherapy, not the challenging patients. Any questions?
Thank you, Bas. I think the only question I see is Randy is wondering whether it was total IL-18 or free IL-18 measured in your study.
The cutoff is total IL-18, and we are also measuring free IL-18. So if there's a difference, we will appreciate that, but the intervention is based on the total IL-18.
All right, very good. Thank you, Bas. So I think we will have more questions at the end, but now I would like to move to the next presentation. Dr. Kimura, could you please introduce yourself?
Sure, I'm happy to do so. Let me just share my screen. I am going to be talking a little bit about some of the challenges of conducting clinical trials in systemic JIA. I am a professor of pediatrics and division chief of pediatric rheumatology at the Hackensack Marine School of Medicine, and I am a co-chair of the CARA Research and Registry Oversight Committee. So I think we all know that the biggest challenge of conducting trials in systemic JIA is that it is a rare disease. There are very few patients. And added to that, it's the variability of the incidence of, wait a minute, can I just try this again? No. Oops, sorry about that. Share screen. Okay. The variability in incidence is in terms of seasonal variability, yearly variability is quite striking, and it's very difficult to predict when patients will present with disease, especially if you're interested in new onset patients. And then there is the problem that there is the availability of already approved products, which makes it more difficult to enroll patients with systemic JIA. I'm going to just give you a few examples of some of the trials that have actually failed to complete enrollment. We've heard about one of them so far, at least the one in MAS. But this has been a pattern that has been a problematic one, and that we need to really try to solve. So ESTIS is a Sobi sponsored trial of Anakinra, and it's a randomized placebo-controlled registration trial, which aimed to enroll 81 patients. This is a study of both systemic JIA and adult onset Still's disease. These are relatively recent onset patients who had active disease. But in the period of time that enrollment was open, only 11 patients were enrolled. And I guess the challenges were really that that Anakinra was already widely available without FDA approval. So, it was difficult to find patients who would be willing to enroll in a randomized trial. And I think that the trial designers tried to make it as patient-friendly as possible with a very short placebo phase, but there were 3 revisions before the trial design was approved, and it ended up being that the placebo-controlled arm was 2 weeks, which made it difficult. And then there was an open label extension that was proposed but was not allowed on ethical grounds because theoretically, since we didn't know what the patient got, the patient may have failed Anakinra if they got Anakinra on the trial, and so they would then just start Anakinra in the open label phase, and that was thought to be unethical, so there was no open label extension. Because of the poor enrollment, the trial was stopped by the company after, I guess, almost 2 years. And despite the low numbers, however, the study met the primary endpoint of the ACR 30 with no fever at week 2. And in fact, all the patients who got drug reached ACR 70, and 5 of the 6 patients who got drug achieved an ACR 90. And all of the patients who received placebo dropped out. Unfortunately, though, this was not because of the small numbers. This is has not yet been approved by FDA and for use in systemic JIA, despite the widespread use in this country and around the world. And Anakinra has been approved by the EU without a registration trial based on clinical efficacy evidence. Rapport was an interesting trial. It was a NIAM sponsored trial that used a novel trial design, the randomized placebo phase design. So this was using a drug called rilonacept, which was provided by Regeneron. And the idea of the placebo phase is that the difference in the patients is that patients are randomized to get the drug rilonacept at day 1 or placebo. And then 2 weeks later, the placebo patients then start to receive rilonacept. And so the actual primary endpoint is the time to achieve the composite endpoint of the ACR 30, absence of fever, and glucocorticoid tapering, which actually the trial did do. But again, because of enrollment issues, there was a plan to enroll 100 patients, but only 71 patients enrolled. It did, as I said, though, still show statistical efficacy compared to placebo. And this is just to show you the dramatic variation in patients who present and are enrolled in these trials. And you can see that In the first year of enrollment, there were essentially one patient enrolled, and then finally in the fourth quarter of that temporal year, 5 patients were enrolled. And some quarters there was a lot of patients, 15 here, and then in other quarters again, very few patients. So this can drag on a study for a very long time and make it very difficult to predict when the study can be fully enrolled. Now,
One more minute, Yuki.
Frost is, thank you, is a CARA investigator-initiated study that was sponsored by Genentech, and also the Systemic JIA Foundation also generously supported the biosample collection for this study. And this should have been an easier study to be able to enroll, because this is an observational study of the CARA systemic JI CTPs. The primary aim is just to compare biologic versus non-biologic CTPs, that is IL-1 or IL-6 inhibitors versus methotrexate or steroids. And we planned an enrollment of 200 patients in 11-16, but enrollment was halted 3 years later. And you can see where it ended here. And even though the trajectory seemed to be increasing, to get to 200 patients would have taken at least a few more years, 3 or 4 more years. So there are many barriers that we had to encounter. Again, the seasonal variability, also the new onset untreated patients are very ill, hospitalized, and difficult to enroll. And also there's a trend clinically for clinicians to really rapidly treat these patients before all of the systemic JIA manifestations have occurred, making them ineligible for treatment. And there's an overlap with stills, anastills. There's also big changes in treatment practice, which I'll show you in a minute. But again, this is just to show you the variability in recruitment for the study, and this is at 60 sites, so it's fairly dramatic. This is just to show you actually how medication choices have changed so dramatically in just a few years. When we did a pilot study of the systemic JIA CTPs, in orange, you can see the distribution was fairly evenly distributed. At least certainly there were more biologic use, but there was definitely still non-biologic use in these patients. And then 3 years later, by 2016 to 2019, when the Frost study actually began, it shifted dramatically, so that 80+% of the patients were started on IL-1 inhibitors. So just to summarize, the very low numbers are a huge challenge. Studying newly diagnosed patients is very difficult. Randomization withdrawal are disliked by patients and parents because the patients are so sick and they're afraid that withdrawing drug or not giving them the drug is going to be an issue. And the long washout period, which isn't on the slide but was mentioned earlier, is also very difficult. And I think that these scenarios and the variability in in incidence makes it very difficult for companies to tolerate. But we have to find trial designs that are more flexible, especially for registration studies. And the design has to be attractive and feasible to patients and providers alike. Thank you.
So Yuki, thank you very much. So what do you think overall, if you look back at these trials, was it possible to tweak something in the design and salvage the trials, or do you think they were doomed from the very beginning?
Well, I think that there were just too many factors. I think the Frost study, for example, if the company had decided to fund the study 5 years before it started, which is when we had proposed the study, the treatment practices would have been a lot different. And we could have learned more in terms of, how patients do on biologics versus non-biologics. But right now, there's really, it's not a comparison that we can do, even in the numbers of patients that we see. I think we'll still learn a lot about the patients, and because these are all new onset patients. So that's the benefit of that. but you know, in the other trials, the Anastil's trial, again, I think if there was a shorter placebo phase, I think that would have allowed more patients to enroll. And again, if there was an open label extension that was allowed, I think that again, more people would have find it easier to enroll.
Thank you very much, Yuki. All right, I think we move on. Ram, I think it's your turn now. Could you please introduce
Thank you,
yourself?
Alexei. I'm just going to try and share the screen. I hope you can see the screen. I'm going to be quick, actually, because I'm going to touch on what a lot of others have touched on. This is the Paracitinib in systemic JIA study. In terms of disclosures, I'm a pediatric rheumatologist in Bristol with a long-standing interest in systemic JIA, but mainly with an interest in novel clinical trial design as well. I'm grateful to Lilly, who are the sponsor of the study, to enable me to present this trial. So I think what I'm going to highlight, this is a study design, I'll come back to it, but you know, these sorts of study designs, when you take one quick look, it straight away doesn't look easy, and it obviously will not be easy. You have to start with a staggered entry by age for a host of PK/PD reasons. Then you have a proof of concept stage, and then you have this, because of which you have an open label lead-in, and the withdrawal design, which we have heard time and time again, is a huge problem to our patient and parent community. It may have a time and place in the late 90s when the first study of a biologic in JIA was done, but its time has certainly gone. It's no longer the time to do a withdrawal design where we give a drug to a patient, show that it works, and then take the drug to show their flare. I think in 2020, we should be, and we could be, and we must do far better. And I'll come on to that in a minute. So the proof of concept straight away posed to loads of problems in recruiting. It's staggered by age group because you want to get the PK/PD data, and we know that systemic and stills are a completely different cohort. But obviously, you may not have the patient you want at the time you want to recruit available when the trial cohort is open. And it's sometimes in the younger age group patients we struggle the most, and they're not available to recruit into the study, even if they're willing. The proof of concept, which was agreed with the regulators, and I'm sure the regulators are coming on later to talk about, was to demonstrate if Paracitinib had a role, which I think most of us believe does, because of its role on IL-6 and other cytokines, on fever and CRP was what they wanted to look at. And also get the pharmacokinetic and pharmacodynamic data. The first problem for recruitment into the study comes with the fact that in the proof of concept period, to be eligible for inclusion, you need at least 2 active joints, and you need to have active systemic features and have an elevated CRP. So in essence, if you have somebody who is partially controlled or controlled on a high dose of steroid, the sort of patient you want to really put to one of these small molecule trials, you have to precipitate a flare. And not only will you have to precipitate a flare, you'll have to precipitate the right flare. You have to precipitate a systemic flare and an articular flare and have the CRP elevation. And forgive me for saying this, but that to me does not sound the way we should be going. And I'm not going to labor the point, but again, you must have at least 2 active joints at screening and baseline. And most of us who manage systemic JIA know that some of our most refractory patients to manage are not the ones who are articularly active, which poses its own set of problems. So I'll touch on what I think, I've been doing clinical trials mainly as an investigator initiated. What are the problems? I don't think in 2020, the randomized withdrawal design is fit for purpose. I would urge our fantastic regulatory colleagues who are here to hear this message from our patients, from our families, and from the clinicians on the ground, that we need pragmatic trial designs which appeal to the patient population, which make drugs available in a timely fashion. We can't have large sample size for a condition such as systemic JIA. The Paracitinib trial is meant to recruit 100 patients. It is an incredible challenge to recruit 100 patients. Why? Because access to Anakinra and IL-6 in parts of Europe make this very difficult. So if you have a patient who's reasonably well, they're going to say, I'll take my tocilizumab and go. If they are refractory, then you can't recruit to the study because you have to stop the medications and make them flare, either develop MAS or meet the rigid criteria to come into the study. And the need for active systemic and articular disease. So I really think it makes life difficult for the clinicians. It makes life difficult for the patients and families for whom the entire regulatory process is working. And in some ways, it makes life incredibly difficult for industry as well, because they have to marry all of these and deliver a trial, which is hard. And I think these are the issues which we can take up with the wider community. And I'll stop there because it's a recurring theme. Thanks, Alexei, and thanks, Vastert.
Thank you, Ram. I think you made a good point. When you try to provoke a flare to make a patient eligible for the trial, I think you put a patient at risk for MAS. And if I remember correctly, in the Anakinra clinical trial, in fact, there were a couple of cases where I believe the occurrence of MAS actually was triggered by the coercion of the patient in the trial. Dr. Brunner, I think you are around. Would you make any comments here?
So when we look at the flare criteria in a randomized withdrawal design, we are talking about minimal worsening of disease and not about a full major flare of disease. In any randomized withdrawal design, patients at baseline are much worse off than those who have a flare during the blinded study. Sure, we would like to have a study design where we know whether the medication is safe and effective without exposing children to placebo. This is possible for some medications and for some compounds, not for all of them. In the very case of a JAK inhibitor, there is no data out from adults where we know how much JAK inhibition one need to effectively treat systemic JIA. To just step back, JAK inhibitors basically block the same pathway as tocilizumab. They block some other pathways too, like interferon gamma, but it's the same as tocilizumab. And we know from the tocilizumab study that children with polyarticular JIA needed much less of tocilizumab as compared to the systemic JIA patient. In order to figure out whether it's from a safety point of view even feasible to give enough Paracitinib to a child with systemic JIA, that's why the fever is demanded in the beginning, because fever is a symptom that quickly dissolves with the resolution of inflammation. It's almost like a proxy measure whether the medication has a chance to work.
Ram, you wanted to respond?
Yes, we haven't
I think Hermina
seen that many.
Hermina makes a valuable point. I don't think the issue is so much the open label lead-in phase. The issue is about the withdrawal phase. So if we take systemic JIA, which is a rare condition, we have effective therapies now, which makes recruitment harder. So we need to target these therapies for our refractory patients. In uncommon or rare diseases where we need the proof of concept, sure, we need to determine the PK and PD, but do we need a withdrawal design? What we need to work with our regulatory colleagues is to see if in an open label design using Bayesian approaches or in a small head-to-head where we assess after the open label phasing, the JAK inhibition versus tocilizumab, JAK inhibition versus Anakinra, so that everybody gets to access a drug which would work rather than put the clinical community and the families in an awkward situation of having to withdraw a drug which has worked.
Well, when you look at, for instance, going back to the canakinumab study, children who went into that trial and hadn't responded by day 3 in the double, there were 2 canakinumab studies, one that used a parallel double-blinded exam design and one that used the randomized withdrawal. In the double-blinded study, if you hadn't responded by day 3 to placebo, you received active drug. By the same token, if you are not that better in the if you get any worse in the placebo arm, you immediately get effective drug again. Now, when you get into this placebo phase, the patient is already significantly better. And in the case of the Paracitinib study, which Printer and the PRCSG designed, you also make sure that the children who get into that withdrawal phase have significantly tapered steroids, and that's important. And even if they taper the steroid, even if they do not reach the efficacy endpoint, so with the JIA 30 at the randomization time point, and the physician thinks, however, that the patient would benefit from the treatment, they will get open label medication. And I think that's a very nice study design. And I would dare to say that, you know, it's more frustrating for family to be treated with a medication that doesn't work. And sometimes when you are on a new medication and you taper the steroids, how would you know in the end that it worked? You need to figure out that it works somehow, and the placebo on background therapy is one way to do that. It'll be great if we had one pharmacodynamic marker where we could say that's the marker you see whether a patient is able to respond to therapy or not, but we do not have that.
Well, thank you, Hermina. That's very helpful. I think we probably should move on. Fabrizio, are you around?
Yes, I am. And while I'll share the slides, I'll present myself. I am a pediatric rheumatologist in Rome, Italy. I have been running a number of clinical trials, and I really would like to start by discuss with you one of the clinical trials that I designed, and then discuss the general principle of the clinical trial for MAS. And I'll get into, these are my disclosures. I'll get into the registration trial of tocilizumab that was published a few years ago, and that led to the approval of tocilizumab for SJIA. Now, when we designed the study, and I'll get right into the debate that was occurring right now, withdrawal design was deemed not to be appropriate for systemic JIA. And the reason why we felt that the design was not appropriate are listed here. We felt that withdrawing an efficacious treatment was not attractive to patients and family. I remind you that the study was published in 2012 and was started in 2008. The first patient was enrolled in 2008, well before the canakinumab studies. And we also were concerned about the risk of flares. And we have been discussing a new, shall I say, USJIA Foundation have been discussing how bad can the flare be in patients with systemic JIA. And although I take some of Hermina's points, I really have to stress a couple of additional points of the risk of flare. The flares may even be worse if you have tapered steroids significantly during the open label. There is another reason why we felt that the randomized withdrawal design was not proper. This is more of a methodological reason. You essentially do the study in responder. Now here below you see the design of a classical withdrawal design with the open label run-in phase. The non-responder drop off. Then you take the responder, the one who have benefited from the drug X, and you either continue them on drug or give them a placebo. If they have responded, they are most reasonably going to flare on placebo. And therefore, the analysis is really biased towards better efficacy of the tested drug. So we really didn't like that. So we went, to tell you the entire story, we went to the FDA and the EMA proposing a randomized placebo-controlled parallel arm study. And we proposed a very short placebo control part, like a few days or 1 week. And there was a lot of discussion, because really the withdrawal design was the trial that was being used in pediatric rheumatology. We kept saying that the JIA was different, and actually they gave the regulatory agency, I wouldn't name which one, gave us a very good suggestion. They talked about escapes. So we designed the study in which the patient were randomized 2-to-1, 2 patients to tocilizumab, 1 to placebo, but they could escape to open label tocilizumab if they had persistent fever for greater than 3 consecutive days, which means that by day 3 they could escape, which is exactly what canakinumab trial used when they started the trial let's say 1 year later. And that has been used in the ANA-STIL design that Yuki has alluded to. And here you see the other reason for escape to open label tocilizumab or standard of care. The advantage here is a reduced, very short exposure to placebo. It's still there, but it's very short, okay? And methodologically, there is a way to compare responders versus non-responder, even at week 8, when it's the primary outcome, based on the initial, it's called intention to treat analysis, technically on the initial treatment that the patient got at randomization. So this is the story of the tocilizumab trial. We can do without the withdrawal design in SJIA, and I think this is an important matter of discussion. Ram, Hermine, and other people has already made their points. Other people will make their points. Now, these are the JIA ACR response rate in the tocilizumab study. There were, let me highlight to you that there were 112 patients enrolled. It was by far too much and too many. If you look at this significant difference in ACR 50 or 70 or even 90, you don't need 100 patients to do this study. You need significantly less patients. Now, let me move to MAS, which is what we have been doing right now. As you know, I designed together with a group of people, including Alexei, the emapalumab trial in MAS, together with the Novimmune team, and now we are running the trial with the Sobi team. Now, the first thing I want to point out to the challenge is the rarity of the targetable disease. We are talking about MAS occurring in, let's say, 10 to 50% of SJIA patients. 70 to 80% of these patients do very well with high-dose glucocorticoids with or without cyclosporine A, as it is being pointed out recently. So this, the challenge here is that you're talking about, if you do your math, 1.5 to 5% of patients with SJIA, which is a very rare disease. Another challenge is the very little data available on therapies in patients who have failed high-dose glucocorticoids. There is no standard of care available for comparisons. We use etoposide, we use T lymphocyte depleting agents. We use all sorts of things, but there's nothing available as a standard of care for comparison. And the third challenge is that MAS is very acute. We heard this today, may rapidly progress and becoming irreversible, with 30% of patients requiring ICU admission. So the randomized placebo-controlled trial in this setting is certainly not appropriate for a number of reasons. We have already highlighted them. The ethical concern, acute disease rapidly reversible. There is nothing to be compared with. The feasibility concern is due to the rarity of the targetable disease. So a randomized placebo-controlled trial here is not a solution, and the only available solution is open-label single-arm trial, which is what the emapalumab trial is. Now, what are the challenge of this trial? I'm not going to tell you about the design. I'm not going to tell you about this. It's an open-label trial, okay, single-arm, so it's very easy. Everybody gets emapalumab. I'm not going to tell you about the results. The partial results have been presented at 2 meetings in the past. 9 patients were, the results of 9 patients were shown. All of them achieved complete response. All of them did very well. All of them significantly tapered glucocorticoids. Now we have enrolled 14 patients. Now the challenge are the following. In well-resourced countries, where this trial is being run, almost all SJIA patients are being treated with L1 or L6 inhibitors. And more often, and Yuki just showed it to you, the frost data, and more often, this occurs very early in the disease course. First line, you get Anakinra or tocilizumab or canakinumab immediately, okay? However, MAS can occur in SJIA patients receiving these drugs, and even when the disease is in remission, well controlled. So the consequence of this is that the great majority of the patients who develop MAS are receiving an L1 or an L6 inhibitor. And therefore, the challenge from a regulatory perspective, but also from a mechanistic perspective, is to dissect the potential confusing effect of the underlying L1 and L6 inhibitors that you cannot withdraw from those of the experimental drug. So how do you tackle this challenge? Well, there are 2 ways to do that. one is the one based on science. We have heard a lot of science on MAS, biomarkers, cytokines, and cytokine storms yesterday. And I think the overall evidence clearly shows that the pathogenic mechanisms involved in active SJIA compared to MAS are different. And you have heard about IL-1 and IL-6 in SJIA, IL-18 is a major predisposing factor to MAS. You have heard about the cytokine storm in MAS, and to all the data pointing to interferon gamma, the target of emapalumab, being a major player in MAS, but not in SJIA. Now, how do we tackle this from a methodological point of view? I think that we use the patient, the proposal is to use the patient himself or herself as her control or his control. Pre-baseline data on MAS activity prior to initiating emapalumab in this case are very informative. Let me make you an example. Now, this is not a real patient. Obviously, I cannot show you real patient data. This is an example of what we might have seen in this study, okay? So this is a theoretical patient with rapidly worsening MAS. The red dots, the red squares are the ferritin levels. While she had SJIA very well controlled on canakinumab and low dose of glucocorticoids, 0.1 milligram per kilogram being tapered. She's slowly creeping to MAS. You see ALT and ferritin going up. She had a canakinumab injection 25 days before full-blown MAS and emapalumab initiation, so well before the 5 half-life requested by methodologically sound, let's call reasoning. The patient receives, when she starts getting into full-blown MS, she receives Anakinra, she receives 3 pulses, significant drop in CRP, which is not shown here, but this treatment failed to control MAS. Ferritin and ILT continue to increase. However, when emapalumab was started, the red dotted lines, there is a rapid drop in CXCL9. You have heard about CXCL9. Yesterday, Scott showed you the data of the emapalumab trial that we showed, that were presented a few months ago at a meeting. And really the change, the decrease in CXCL9 preceded the changes in the mass MAS parameters. Here again, you see the drop in ferritin and ALT. And there is a progressive tapering of glucocorticoids and shown in the box below the graph. So this is what we can do in open label study. And there will be a novel study with emapalumab, hopefully, that will really be based on a run-in period in which we collect a lot of data on the activity of SJIA and MAS prior to initiating the experimental drug. The other issue with open label trial is the absence of a comparator arm and the absence of a statistical hypothesis. And I think here, importance of natural history studies comes into play. And we really have to work hard, particularly in the field of rare diseases, to build natural history studies together with parents and patients, because I think this will make a difference, and is going possibly to make a difference. And even more though, in the setting of interstitial lung disease in SJIA, we really need pre-baseline, long-term data, possibly high quality, on the course of the disease. And this data and the quality of this data has to be agreed beforehand, because it's complicated to collect, has some privacy issue that have to be tackled. And really, the quality of the data has to be satisfactory for the regulatory agency to approve this treatment. And I guess that pre-baseline data and natural history studies here in this specific setting, which is so close to the heart of this group of people, are very important. And with this, I finish my presentation, and I'll take any questions.
Thank you, Fabrizio. Hermina, you have a comment, correct?
Oh, with respect to the emapalumab, well, it's basically the secondary use of an approved medication in a very similar disease, and so the situation is different than for the Paracitinib study, for instance. But whenever I think study design doesn't always really depends on the type of the medication and how much we already know. For some medication, an open label study is sufficient because we already have a good knowledge about the dose that is sufficient in other children with a similar disease. And here we would have the emapalumab, or sometimes we already know we don't have that knowledge. For instance, the canakinumab study. So when we look at the study design, it's really dependent on what we know already in a children, in adult, how severe the disease is, and how feasible the study is.
Can I answer?
Sure. Yes, go ahead. Yeah.
I disagree with you, Romina. Kindly disagree with you because we are good friends and you're smiling, and me too. But really, MAS is a different story from SJIA. I don't think there is no issue. When we started the SJIA trial on emapalumab, there were 10 patients treated in the primary HLH trial with emapalumab. So this is not the case. It's not a secondary indication. Okay, so this is not true. We started from scratch, and Alexei knows how difficult it was to think about how to design this study and what to do. Regarding trials in SJIA, I do believe that the PK-PD approach, PK, because we don't have a PD marker. We don't have a PD marker because PD marker were never studied for some drugs. Let's make an example, JAK inhibitors. They were never studied. Nevertheless, while in the emapalumab trials, the PD marker is there always. So in the PK-PD approach is mandatory in children. And the age staggering that Ram was painfully alluding to is somehow, needed. Makes things very complicated. I know, I see him nodding. Make things very complicated from a feasibility point of view. But I think this is needed from a methodological and safety of the patient's point of view. Now, then we disagree. I mean, then it's a matter of how to approach SJIA in a setting where IL-1 and IL-6 inhibitors worked very satisfactorily for 80% of the patients. I hate to say, actually, we don't need anything else for this 80% of the patient. We need VAS trial, the ESTIS trial, to understand who's going to withdraw the drug. Okay. Then Paracitinib should be, and other. We don't want to name. I mean, do we have an issue with Paracitinib? I don't personally. So any other should be tested in the non-IL-1, non-IL-6 responder. I see Ram eagerly.
Alexei, I just want to add a comment I completely agree with Fabrizio, and the age group is a painful necessity. And I also think that the withdrawal design is where I have an issue. I think once we have proof of concept, we can either have an open label, given that this is a set of patients with the refractory, and like Fabrizio absolutely correctly points out, the need is for a tiny number of patients. And if required, we can have a reference arm of patients real-time treated with IL-1 or IL-6. It's the withdrawal design, I think, as a pragmatic on-the-ground clinician, I have an issue with in 2020, although it's served us extremely well when the biologics first hit the ground.
There's lots of designs. You can do platform trials. You can, now we have better historical data, but at the time when the study was done, that was the only, way I see that could have been done scientifically well. I need to go back on the PD. We get stuck on the pharmacokinetics, but truly what we are interested in is the pharmacodynamics. And as per a recent workshop at the FDA, it's really pointed out that we don't really focus as much on that part. We just assume that if we give children the same dose, the same drug exposure than adults, that it miraculously works in the very same way. And we do not even entertain at the current time whether a higher dose could work better. We do it in clinical care. And, Randy has all the experience on that with the use of Anakinra, for instance, showing that the doses that are approved for adults are just not effective for children.
Very much agree, Hermine. And I think that the PK data, also PD data, is also very helpful to link the effect to the drug. The example that Fabrizio showed CXCL9, something that measures interferon gamma activity, right? So he clearly showed that CXCL9 was going down with the treatment, and it was closely followed by the improvement in the MAS clinical parameters. So this kind of temporal relationship between the PD parameters and the clinical parameters of the disease, I think could be another sort of argument showing that the effect is indeed attributed to the drug. I think it's a very good point. Well, I think thank you very much, everybody. I think it's time to move on, and I think it's time to listen to hear the perspective of the pharma industry, because again, that's the other side of the story, and there is a separate set of challenges. And I think we will start with Dr. Waldron-Lynch. Frank, are you there?
Yeah, I am, Alexei, so nice to speak to you. Just to introduce myself, my name is Frank Waldron-Lynch. I'm a translational medicine expert based in Novartis Institute of Biomedical Research in Basel in Switzerland. So, I mean, I'm really principally here just to listen, to understand essentially the patient journey and really the challenges that you're experiencing in terms of development in this area, as well as potentially the opportunities for development. And everything that I've heard, Fabrizio, Ram, Alexei are the same type of discussions that we have internally when we look at SJIA or other rare diseases in terms of trial design, population, and then I think the last comments about establishing early relationships between PD and also PK-PD relationships are really essential really to drive development forward. So I'll hand it back to you, Alexei.
Thank you, Frank. So I believe
Eduardo
is here too, correct? Eduardo, could you please comment?
Yeah. Thank you, Alexei. Thank you, everyone. So yes, in fact, the meeting has been so rich, and for us is a source of discussion and knowledge, as the previous speaker said. We understand the value of the biologics and how the biologics have improved the management of SJIA, and we have this 80% or something like that of efficacy and change of the clinical course of the disease. We are interested though, and we are interested in the IL-18 blockade pathway of the diseases. We are interested in a fraction that still remains with an unmet medical need. And this is a subpopulation of SJIA patients that are lacking a response to the treatment, including the biologics, that they have acute and chronic complications like macrophage activation syndrome or lung disease. And for us, this is really a place where we would like to explore with our treatment. Not only, I think a treatment that could fulfill the control of inflammatory activation of the disease in this fraction of patients, and at the same time prevent the complications, the acute and the chronic complications, would be something very valuable for a group of patients. And this is where we are discussing. It's a very challenging. So we have the evidences from the literature and also from our own clinical development plan of the value of IL-18 binding protein, recombinant IL-18 binding protein, in the control and the management of HLH macrophage activation episodes in monogenic autoinflammatory diseases, and also in the control of Still disease. So the question is, moving forward, if we could really identify and reach a population that would benefit of the IL-18 blockade in this field. And of course, we have learned, I mean, I have learned a lot during the meeting, and we have seen that it's difficult to find predictors. How do you really identify a subgroup of patients that could be of benefit for these patients? So for us, we tend to think that the high IL-18 levels have, Scott kind of talked about the mass, and he gave some cutoff levels. And we also saw today from us the levels of IL-18. So the levels of IL-18, the systemic inflammation, the early onset, the history of recurrent mass, smoldering mass, or this persistent partial mass. So we don't know how important this group could be, but we feel this group could benefit from the IL-18 blockade. And this is very challenging. First of all, the population will be even more reduced than the population that we have been discussing so far, because we are selecting a subgroup of patients. Also, if we want to prevent some of the chronic complications, the length of the study will be longer. We don't know if you, Alexei, with Schuler, you have identified surrogates, early surrogates of the complication, of the chronic complication like lung disease. This will be very important for us. So these are where we are. We know we can treat long-term patients with a very safe profile. We have now in the compassionate use patients that have been more than 5 years with tadekinic alpha without severe adverse events. So we have this value. We think there is some sweet spot to identify a subgroup of population of patients that would benefit from this treatment. And this is what we are doing. The enrollment, the design, all you have said, all we have learned will be very challenging. And we will be discussing with the FDA. A final thing for the authorities, I think, to consider the design, to consider the difficulty of running a trial in COVID times. We have a hard time to finish a phase 3 clinical trial in monogenic conditions, and it has been very struggling to enroll patients, to complete the protocol and the visits in this time. Patients and parents that have to travel from one side to the other. So this is very difficult. We are doing all the effort to complete this. And a final acknowledgement to the parents and patients. They have respected the protocol, the integrity of the protocol incredibly. The investigators, we have had a lot of support from the investigators, and we hope, and we are discussing actively with the authorities in the US and Europe to see how we can move forward. But this is where we are, and we are interested in probably in this subgroup of SJIA patients that could benefit of an IL-18 blockade and prevent chronic and acute complications. Thank you. I went too far, right, or too long? I don't know.
Thank you. This was perfect. We also would like to hear the perspective of Sobi. Ravi, are you around?
I am, Alexei. I don't know if you can hear me. So thanks very much for asking us to the meeting. And I just want to echo what everybody else has said, that it's been fabulous to listen. So I'm representing Sobi. I'm the head of R&D. And obviously we have been involved in studies with Gamifant and with Anakinra that have been mentioned extensively. So I probably don't have to reiterate some of the challenges and frustrations we obviously have felt. I do want to acknowledge all of the effort that you as investigators and parents and patients have gone through with some of these trials. It's particularly striking hearing the story earlier on from New York about traveling to Cincinnati for the emapalumab trial. Obviously, I don't want to reiterate the rarity, the RCT and trial designs, etc. I think just to add on something that's really beginning to kind of attract our attention, and particularly in COVID times with the interest in cytokine storm and the overlap between clinical presentation of HLH and cytokine storm syndrome was an article that I think one of you may have written earlier in the year, whether we should lump or split in these very acute scenarios where the phenotype may end up being quite similar, the pathology and pathogenesis may be quite different, and how do we therefore treat it? And obviously, which cytokine should we use, or which inhibitor should we use? And I worry going forward that it's going to be very difficult for us to delineate the individual diseases, and how will we do that? Depending on the etiology of cytokine storm or MAS or secondary HLH, and how we will define those. I'd like to echo the suggestion from Fabrizio, and maybe it's a plea to you and then to those from the regulatory authorities, perhaps really strong natural history data to act as a control for SJIA outcomes and mass outcomes would be the right thing to do, because I think all of you probably recognize that in a child or young adult with macrophage activation syndrome, there's a huge reluctance, understandably, to change any medication or to withdraw any medication. And that becomes difficult, I think, for people to correctly interpret the data. And that's something that we've definitely struggled with. There's a question on the chat that says, is it true that the EMA have refused approval of Gamifant for HLH? And the answer is yes, they have refused approval. It's the same data set that's been approved in the US. And Fabrizio knows the trial design better than anybody. And I think that we've really struggled to be able to argue that you can determine an effect of emapalumab in those patients, different disease, same principle. So I think we would like to say, look, we need to be able to judge these drugs in the same way, using sort of synthetic or natural history controls. And maybe a plea to regulatory authorities is to treat data in the same way when we're talking about ultra rare diseases that are hyperacute. So that would be the perspective that we would have. But I would really like to echo a lot of the things that have been discussed already.
Can you comment on the possible use of the PK/PD data as a sort of another way to link the effect of the drug?
So for Gamifant, we've looked at CXCL9 in particularly as a biomarker of the PD effect of emapalumab, simply because it's so much downstream of interferon. You could look at CXCL10 as well. And I think we've got relatively strong data. It's a difficult thing to measure, so we are embarking upon developing, a more widespread and robust test for that. But I think there have been mixed reactions, both from the FDA and the EMA, on utilization of CXCL9. I don't know how many of you use it in your clinical practice and how robust it is. So that's one thing we've done. And the other area that we obviously have collected a lot of data is on the laboratory parameters of mass. And again, I don't need to educate this audience. Some of those are tracking with different temporal relationships to maybe a clinical effect, and therefore ascribing a PD effect in that regard has also been a little bit challenging. But I think it's something that we probably have to be able to do in a single arm, uncontrolled setting, potentially with concomitant medications that could be very varied. I think Eduardo mentioned it, as did Frank. I think, we should accept that a PD marker should provide, very strong corroborative evidence as we're looking for evidence of efficacy. But again, I'd be very interested to hear what, the experts would say.
Thank you, Ravi. I don't think I see any questions to you at the moment, but probably we will have a later discussion and it may come up later. Thank you, Ravi. So let's move on to the next stage in our discussion. And this time we would like to hear first the FDA perspective on all these issues. Nikolay, are you around?
Yes, I am. Alexei, do you hear me?
Yes, perfect. Thank you very much. Thank you again for joining us. And again, you are a pediatric rheumatologist, I believe, by training, right? So you should feel the pain of our patients.
Yeah.
So please tell us what you think and what's your view of all these issues.
Sure. So first of all, I want to thank Rashmi for keeping us engaged and involved from the very beginning of when she started running this incredible meeting every year, if not almost every year, and giving us this opportunity to interact with this community and with you as sort of the key leaders in this field, the investigators, and also with the industry stakeholders. I think this is an important opportunity for us to hear what patients and parents feel and think, and also what are the challenges in that area. And I think we fully recognize the challenges with managing systemic JIA and the complications in the MAS. And I think we understand also the disease enough that we can use that knowledge for driving or directing drug development in that area. I think FDA and I think the regulators play a certain role in all of this process to make sure that drugs are properly tested for safety and efficacy and make these products available to patients. I think we have shown interest in being open to pretty much any suggestion that comes to us that would answer the questions that we have, which is whether a drug is safe and effective. But there are many other factors that were discussed today and what we heard from patients and from investigators and from the industry that are in play here. So I think we have to navigate all of this difficult landscape in our considerations. I think, I'm not really sure which parts you want me to address specifically, but I think that we had a tremendous success with our interactions with the community, with the larger community for JIA in general. And we kind of built on that experience to have a very robust scientific discussion last year. And many of you on the call here or the WebEx here have been a part of that discussion, which we think provided a lot of insight into how we could or should approach drug development for JIA, not specifically for systemic JIA, but I think many of the principles apply to systemic JIA. I think we took this to heart and we actually applied the principles that were discussed at that workshop in October last year for trying to expedite development and approval of some products already that we thought was scientifically justified based on these discussions. And one example would be the approval of Anakinra for Still's disease based on the discussion of the PK-based extrapolation. We thought that that would be a reasonable approach and based on the understanding about the similarities between pediatric and adult patients, we thought that was again, a reasonable approach to do so. I think we kind of learned from that experience and we applied it to what we do. We wish we have more of these situations where it would be easier to apply this approach. We also heard that last year in October at the workshop and also today at the discussion that there's significant challenges between transitioning pediatric patients to adult care. And some of it, applies to drug development and how we view these diseases. So trying to align adult and pediatric rheumatology fields as much as possible would make it a lot easier for us also to think about how we can leverage that scientific knowledge to approve products and make them available for both pediatrics and adults. I'll pause here and see if
well, I think one question that we have on our side is natural history studies were mentioned several times today by pretty much all the sites. So what's your philosophy? What the philosophy of your agency on the value of those kind of studies?
So, I'll speak for the FDA at least. I think what we need to make sure is that new products that we approve actually work and they're safe. And one way to demonstrate that they work is, the traditional placebo-controlled trials, which, is probably easier said than done in this setting. But we still need some sort of controlled comparison. And if we don't have the, concurrent control in a clinical study, we're looking for some other ways to account for that control. And control could be a historical control or control from natural history. if it's well understood and well characterized. And if we feel comfortable that at the end of the day, we know how to compare any efficacy that's assessed in a study versus the natural history of the disease. So I think this is critical for us to have this information and to consider it's if we cannot really do a concurrent control, whether that's placebo or some other active control study. So I think the agency is open and has certainly considered this. In the field of oncology, many products are approved based on single arm treatments protocols because the natural history is so well characterized. The tumor doesn't shrink or the tumor doesn't go away. So it's much easier to compare results in a clinical study to that historical control. So if we have that level of confidence and information about the natural history of a disease, we would certainly consider that without the need for a placebo-controlled study.
I can add a comment.
Sure, sure, Rashmi.
So the foundation is actually, along with a network of researchers, we are launching and working on designing a natural history study. So this is very relevant to us, and and we definitely want to do something that will end up helping with these kind of issues. And if you have, we can do that even in a follow-up, examples of diseases which are relevant to us. I mean, oncology is relevant in some ways, but on the other hand, the diseases can be characterized much better, like about a tumor, than what we can. So, but if you give us examples of natural history studies that did help serving as good controls, then we can take those and make sure we go through them, look at those study designs, and do something that can actually help us move things along. Do you have, can you think of drugs or diseases that are relevant to, which have natural history studies and which are relevant to us?
So I don't think I have examples other than the field of oncology. Again, actually, I can reference maybe the field of vasculitis and the approval of rituximab, the initial approval of rituximab, where the approval relied on comparison to historical control data. So that was a non-inferiority program for non-inferiority study versus Cytoxan, which was a standard of care at the time. But the assumption was that if you didn't treat these patients, you had a very clear picture of how worse they did or whether they died. So I think that's another example in the field of rheumatology where historical control was considered as part of this assessment of efficacy.
Okay. Are there any? Sorry, go ahead.
No, I think the most important question for us is how well characterized this natural history is. For example, if a disease has a fluctuating nature, if it waxes and wanes, then it makes it a lot harder to assess whether improvement that you see, in a single arm study is because of the drug or because of the underlying, disease was fluctuating. So this is certainly a lot more challenging.
Thank you, Nikolay. I think now we probably should move on to and listen to the European view on this issue. Richard, are you here?
Yes, hello, can you hear me?
Yes, excellent, thank you. Thank you for joining us.
I'd like to thank for invitation to this important meeting. It's good to discuss all these points, and there are so many that it's difficult for me to decide what similarly to Nikolay, what should I address? On top of it, Nikolay already explained most of the things that we discussed together many times, and we have actually very much converged our positions during the last years. Still, we have some differences, of course, and there is a criticism from Sobi for different outcome. I just remind that different outcome was also for other cases like Anakinra or golimumab. But I think we converge a lot in understanding the data and the need. However, we still may have different outcome, which is a result of the scientific discussion. There was a question whether money played any role in emapalumab decision. No, absolutely not. We don't even know the price, and we don't take into account anything except science. So maybe just, I feel humbled to address a number of points that such outstanding experts as you have already expressed their views, but I still feel there is some discordance between views of experts. Maybe I start with just saying the basic thing that what is our role as regulators? Our role of regulators is just to assess whether the drug works and whether it's safe. And that is really the question that we are addressing. And there must be someone who applies to regulators with an application for the drug. So many times there are investigation-initiated studies that don't have the ambition or tools to get to the registration. And that is something that is very important, that and I would really apply to investigators to have this in mind, that the study one day might be used for registration or as a data source for evaluation of the benefit and risk, because this is many times not the case, and exposed many times these studies are not usable. And we see it now in COVID-19, for example. There are a number of studies initiated by investigators that are very small, too small to show really anything. The next thing that I would like to stress is that we, of course, base our outcome on the evidence. So the clinical studies, but it is not only clinical studies. It is also experience, including small studies that appear in the literature, and many times also kind of thinking about the data, what we call extrapolation. And that is coming more and more, and we discuss a lot about these things with FDA. For example, in golimumab case, we were able to authorize golimumab based on existing data about this drug. And I agree with Hermine that it depends very much on the drug and how extensive is the knowledge outside of the existing data in children. I'm not sure if I'm still with you because I lost your picture, Alexei.
We can hear you.
We hear you, we see you.
Thank you. Now, as Nikolay said, we are open to many different sources of data. In our guidelines, for example, for juvenile idiopathic arthritis, we list that we are open. Of course, randomized controlled, placebo-controlled study is the golden situation when we clearly see the benefit. But we also understand this is not always possible, and we list also other designs and other sources of information which we are open for consideration. And as I mentioned already, for example, in golimumab case, we do it. And then comes the outcome when we have to compare benefit and risk. And here patients play an important role, and I'm very happy that you actually in United States have this organization organized by Rashmi that is so powerful and able to produce positions. Because oncology was mentioned here. For example, in oncology, there is a great example of individual risk for patient. There is very different situation in different oncology patients, and also individual acceptance of risk. In health-threatening situation is very much different acceptance of risk than in much less threatening situation. And many times we deal with individual patient views. So it would be great if you, as an organization, could somehow come to us with a clear view what is acceptable by patients, what is the need of patients. I don't think you are there, because I hear different views and read different views from different patients. But for our decision making, we are open to hear patients, but many times we don't hear the clear message from patients, what they really would like to try the new drug independent from the risk, or to be more careful and really accept only medicines that don't have any risk. So I think that is more or less what I wanted to say. So what is the role of placebo versus single-arm trials is a very difficult question, and we discuss it round and round for years. And also during this discussion, we heard different views. I think this is not the question for regulators. We, of course, have much easier life when we have placebo-controlled study. We also understand it's not always possible, and as Nikolay said, we have number of, especially in oncology, number of products that were accepted based on only single-arm study. So we are open, but we need, and I very much welcome what Fabrizio outlined, the most possible clear data that would allow make some conclusions on the single-arm study. So that is something that is very important in preparation of that study, keeping the population as clean as possible, allowing isolation of the robust treatment effect, that in that case we would be able to accept single-arm studies, and we share all the concerns about use of the placebo. I think Jan is here also with us, I hope so, who might have much more useful comments to the design of studies than I have. I forgot.
Thank you,
but I hope you know me. Jan is member of CHMP and also Scientific Advice Working Party, very much dealing with rheumatology products. He was chief head of Rheumatology Working Party at the agency. So I hope, Jan, you are with us.
If I may, I just want to say something. I love this concept of the individual acceptance of I wrote it down. I want to make sure, individual acceptance of risk. I think I did not know about this specific concept, but in my own mind, I keep on thinking, I know what my child is going through. I know, I know the risks and benefits. I sit down and discuss them with my doctor all the time. And I know there's a bunch of patients like me, but we have no avenue to say this to anyone. We just are dependent on these clinical trials that are for different scenarios than our scenario. And that's really what we are trying to say. I want to understand what this means. We have to put together maybe a bunch of patients and say, this is what risk we are willing to accept. And maybe there is something like this for the FDA. But I think this is the important concept, and we take this feedback to heart, and we'll go back and research this and try to figure this, what we can say as a patient group. Okay, sorry for that. I just wanted to make
Yeah, so Dr. Müller-Berghaus, I think you are another very important voice in Europe, so we certainly would like to hear it here.
Yeah, thank you very much. Yeah, it's very difficult to be in the last spot when so many people have already spoken who are much more knowledgeable than I am. So just as a background, as Richard mentioned, I'm currently at CHMP and I have an interest in rheumatology. And I'm also the rapporteur for Roactamra and Ilaris. And that sets the scene in a way that I'm very, very much interested in randomized trials. I am so happy that we had randomized data to look at and base our decision on, because this is a robust decision making that we were able to do. And I think in the end, able to provide patients access to this important drugs. I fully recognize that with the availability of drugs and availability of different treatment options, it becomes a lot more difficult to design the right trials or to design trials that are able to tease out efficacy and safety for the newer compounds that come onto the market. So the oncology setting was mentioned, and I think that one particular topic in the oncology is that if you look at the approvals in single-arm trials, you start essentially at the last line. You start in the most desperate situation with single-arm trials. In addition, you have the possibility to judge the effect of the drug based on response rates. I have to admit that in Europe, we're having lots of discussions and indeed very controversial discussions whether the response to the tumor actually already indicates benefit for the patient. And similarly, I think you could transfer this to the setting of SJIA, is what is clearly a benefit for the patient in a desperate situation? What is the benefit for the patient who starts to run into MAS? Can you define unequivocally, without any doubt, something that would indicate the benefit to the patient? And I think if you are able, as a researcher, as a physician, as a mom, to tell the regulator, this is for me such a clear indication of benefit, I think regulators could easily live with single-arm trials. And for me, that is really the crucial point. First, you start you define your population. What are you interested in? What is sort of the therapeutic setting? Is there anything available in that therapeutic setting? How much is the patient endangered? And then you decide on what is my capability to capture that benefit. And I think if you've done that, then you can start thinking about what is the most appropriate trial. We have used single-arm trials, especially in genetic diseases, where it is clear how the genetic diseases run. It is clear that, for example, hemophilia will not cure in itself. You will not get a normal blood coagulation system in a patient who has a genetic disease such as hemophilia. There will be no waxing or waning of disease, so that the clinical course is very clear. If you're able to find that for your specific population, I think we would more than willing to accept that. The examples that we had in some rheumatology-related disorders is that the patient population was poorly defined. It was essentially left, for example, to the physician to decide what was severe disease. So you need more objective criteria that you could use then to come to a conclusion that this is a certain spectrum of the disease where you could do your trial. On the role of the external controls, I think I really like what Nikolay had to say there. It is a very important data for us for the reason I've said. We're using single-arm trials and we need some reference point. So in a setting such as hemophilia, you have the reference point, the patient himself. He will have had bleeds because he doesn't have the coagulation factor. If you are able to get this corrected, he will not have bleeds. So you can compare the before and after. If you have a patient with MAS and you are able to define for the patient a sort of a uniform course, then you could compare before and after. But this, for you as the experts, to tease out or to define that's not something that I as a regulator can do. I can look at your proposals, I can try to probe that proposals, but I think the initiative has to come from the investigators or from the patient side, defining that course. The other possibility for an external control is of sort of historical control or a concurrent control where you collect data within a registry. And then this is probably more addressed to the industry. You need to start early to think about this. It is too late thinking about this when you're starting your trial. So once you start developing a drug in a certain indication, you should already start thinking about how do I collect here my natural history cohort. Yeah, so you have enough time to think about how best to do that, what would be the criteria and parameters that you need to collect. It is way too late to start that if you are already in a pivotal phase when you're going sort of to the final stages of an approval. The earlier the better. Then you can still collect concurrently to the trial to patients who have not access to the trial for whatever reasons. But thinking about this is important because it takes so long. We're having experience with established registers that people want to use as an external reference or an external control. And we find out that many of the things that the developers and we are interested are just not collected in that register. So nobody has thought about collecting that specific parameter, and then you cannot use it. So the thinking has to start earlier. Rashmi, if you come with a proposal for a cohort of SJIA patients that you think would be useful in the future, this is something that we can discuss in regular settings. We have meeting formats for that, where you would say, this is actually, we propose to use this cohort or this way of collecting and these parameters for collecting to characterize a patient population for future external controls. And I think in Europe, we would find ways to discuss that with you because we recognize how important that type of discussion is prior to an actually decision making. Because if you do that without ever talking to anyone, and then when a company comes and says, oh, cool, look here, this is our comparative data, then it's too late. It's way too late. So you need to establish sort of interactions and discussions. And I'm very happy to be here today and really understand the problems. I think I have a lot better insight now. Whatever I said is not new. All the others have said exactly the same things, but you need to push for dialogue and acceptance early on, I think. I don't have to emphasize the need for good trials, especially if we can get placebo control. But I fully recognize that there are other ways and there should be other ways. And I think we have proven that we can use other ways.
Well, thank you, Jan. This is an excellent point. I think Nikolay had a comment. Nikolay?
Sure. Thanks, Alexei. So I really wanted to build also on the comments by Jan and Richard. And I think we started talking about the external historical control and alternatives to a placebo-controlled study. Kind of understanding the placebo by itself is problematic, whether that's prospective placebo control or randomized withdrawal to placebo. Both to patients and community. So we have to consider these issues when we advise sponsors for designing clinical trials. But in the space of systemic JIA, I just want to bring another point maybe for consideration. We have several products approved, and we are certainly also open to have active control studies, which should address all of the concerns about undertreating patients or exposing them to placebo unnecessarily. Granted that these might need larger sample sizes, but recruitment should not be an issue, and that would certainly address many of the questions about assessment of efficacy and safety for new products. So I just want to put this out on the table to hear what people think about this approach, rather than us trying to rely on uncertainties from external controls or from the disease itself, which is not well characterized. I think this is one area where I think we should consider these as an option.
Fabrizio had a question.
I still have to learn to unmute myself after 6 months. I should do better. I had a question for the three people from the agencies on the definition of unsatisfactory response / resistant disease. Now, my experience, particularly with biologics, is that let's talk about rheumatoid arthritis trials, for example. The definition has always been left to the treating physician, with a few thresholds in the disease activity level that the patient had to have, number of active joints, CRP, and in SJIA, we can certainly borrow from that field. It's a bit more difficult to do so in MAS. And although we, for example, just to talk about what we are talking about, the emapalumab trial, we are enrolling in the trial patient with failed high-dose glucocorticoids. There is no, what are your suggestions on the definition of unsatisfactory response? And should we include, I mean, ideally we would include the parents and the patient themselves in this definition. However, this is a very acute setting, a rapidly evolving patient that may become acutely ill in a few hours. And it's a very difficult, sometimes, can I say, a request to define this more strictly? I don't know who wants to take the question.
Yeah, I mean, I'm not the expert, so I can start and leave the but that is the question to you. You are the expert. Are you, if you are not able to define what is a relevant response, how can the poor regulator like me, just a pediatrician, run-of-the-mill, not rheumatologist, decide what is good? So it should be you coming with a proposal and discuss this with us, and we can then dive into the literature, discuss amongst ourselves, discuss with parents, discuss with representatives, whether this would be something that would be worthwhile considering. I think it's a bit like ACR. I mean, that's not something that regulators invented. That's something that came from the treatment community, right? And then it sort of moved into the regulatory field, and we accepted the criteria that pediatric rheumatologists developed. Sometimes we were a bit unhappy, but okay. But in the end, I think it is the experts who have to do the work and come up with something. It's not I don't think it would be appropriate for me as a regulator to impose something on you where you are the expert. But if you come up with something as the expert, it should be robust, and you have to make your point and demonstrate that it's robust.
I think Hermina has an opinion on that too.
Well, I agree with that sentiment. We need to come, the experts and the people who are familiar with the disease need to come up with the outcome measures, and they also need to be involved in the study design so that the study designs are sensible and can be tolerated by the families, and that we have valid outcomes. Example, like the tocilizumab study. Fabrizio was very involved in the design, and that was and with the outcome measure choice, and that was important for the success of the whole program.
If I may say something.
Yes, please.
I don't see the hand up button, sorry. I just want to say, Fabrizio, there are a lot of examples in rheumatology when the line of treatment is being defined in the indication. If you are asking about specific situation in acute disease, exactly as Jan said, you have to define that. What is important is that the population that you end up with is clearly defined, and that the data come from the patients that are homogeneous, that there is no so big heterogeneity that this allows really making any conclusions in the line of treatment. So I am not sure if I understood your question. You are so experienced that you probably have something in mind that maybe I did not get. But in principle, I think when evaluating results, it's important, and speaking about line of treatment, that we understand that we are having data from the homogeneous population.
Thank you. We have a few minutes left. Any other comments from anybody else?
I have a couple comments, Alexei.
Go ahead.
One being, I think it's number one, going to be really hard to get homogeneous patients for macrophage activation syndrome trials. I think that's going to be close to impossible. But I do think there are a couple trial designs that may be beneficial. And one may be just comparing dosing of agents, one dose to another without having a placebo. It's a possibility. I do think one of the things we may have gotten at least partially right in our Anakinra trial for MAS was giving both arms steroids. And maybe the trial design is steroids in both arms, placebo versus your drug, and then tapering steroids, and then being allowed to switch into whatever the drug may be or something else for that matter. But if steroids aren't helping you, particularly at high doses, you're in trouble. So steroids are most likely going to help them. So at least we're not putting the patient at extreme risk, other than all the lovely things that come with steroids. But in the short term, you need to get the MAS under control. And almost everyone uses glucocorticoids in that setting. So that's, I think, one thing that we may have partially gotten right in our trial design that may be important for future MAS trial designs.
Thank you, Randy. Anybody else?
I have one more point I'd like to make in the context of this ethical difficulties, that you have a drug which seems promising and you're not able to randomize anymore, and it would not be accepted by patients or carers, families to go for something that they perceive as so promising. So I think this is also a call to industry in a way that you need to think about randomization much earlier in clinical trial. If you are with a targeted drug that really has a good scientific basis and you start out with dose finding trials and then looking around where this could actually go, I think it is a good idea to start randomizing as early as possible, because at a later point in time, you may not be possible to do that anymore because it's perceived as not ethical. And for regulators, it's extremely hard to ask for these randomized trials. It's not something that we want randomization desperately, because it's the best way really to define efficacy and safety of a drug. But we recognize that at a later point in time, it is so difficult. So the consequence for me would be try to do it as early as possible and think about possibilities, not wait too much, because I think the term equipoise is the right one here. If people don't believe in equipoise, also on the investigator side, you will never get this trial running. So you need to do it earlier in a way, and think about possibilities of doing that.
Thank you. Rashmi, I think we're out of time.
You can take a few minutes if there's I mean, I don't really have much more of closing comments.
Any other final comments?
Yeah. I had a question, I guess, for Nikolay, if that's okay. I wanted to know, is there any avenue for us as we try to design this natural history study to engage with the FDA to kind of make sure we get it, design it right, we think about it the right way to be able to use in future clinical trials as controls?
So what exactly do you think of?
Well, I mean, Jan mentioned that maybe we could have some conversations even at this stage, at an earlier stage, so that we get it right from the get-go. So I was asking if similarly, if we can engage with the FDA, so I come and say, this is what we are planning. And this will be for a future MAS or a future SJIA trial. Because we are just now at the stage that we are planning it out, so I want to get it right.
I guess one thing that you can consider is maybe to talk to the other registries and how those were set up. That can be used also to characterize natural history. I'm not sure whether CARA might be, one example that you can use, or at least talk to them about how they have done that. I think from the FDA side or the regulator side, it would be more to ensure that the data are collected reliably.
Okay.
I think that's really a key for us, so that we can look at the data later on and say, yes, we can trust that data for whatever purposes. I think we'll be open to provide any advice, but I'm not really sure that we can drive that decision ultimately.
Fair enough. I meant more like, should we do, there's a bunch of different designs. I'll just send you an email and follow up and see. We are not looking for much. I just want to make sure we don't design the study a different way than it should have been. But I think I get your point.
By natural, well, I guess we can maybe talk offline about something like this. And I just looked at the chat. There were quite a lot of concerns expressed about the proposal to do active controlled studies. So I think we have to pick and choose our battles, and if we don't think that placebo exposures are appropriate for this patient population, what are the alternatives? So I think we should not dismiss active controlled studies upfront without thinking through this question, and we believe that these could be possible to do without exorbitant sample sizes. As there was a comparison to rheumatoid arthritis field. Again, we're talking about different effect sizes for different disease. So I think that's one option that needs to be considered. And I think this is certainly going to address many of the concerns expressed by patients and parents, and may provide a lot of useful information for both safety and efficacy of a new product. Because I think the bottom line for us is that we want to make sure that a new product is safe and effective. We should not assume it works, even though in anecdotes and in certain cases it will work. And that's why it's in development, because it has shown promise. But we want to make sure that we can, quantify the benefits and the risks of a product at the end of the day, again, for approval and for making this available to patients through this pathway. And, having a product like Anakinra that is currently standard of care makes this assessment somewhat challenging. Again, I just wanted to share these thoughts from our perspective. I don't know how EMA might think about those, but I think these were important.
I think we can agree with you, Nikolay, and I just wanted to add one thing. I'm physician by training, pediatric rheumatologist, and when I came to the agency, it was very difficult for me to accept this different view from the clinician to the regulator. What we are trying to create in regulatory requests is indeed an artificial situation, very much controlled and maybe even non-existing in real world. And that was very difficult to accept for me. So I take Randy's point that homogeneity in such a situation as a rare disease is almost impossible. But we have somehow to find the common ground to allow really to find the difference that medicine does. So what I'm calling for is to meet somewhere from both sides that I live actually in my person, somehow to find a compromise that would allow regulators to really isolate the drug effect, to define the population as much as possible and to really distill somehow the influence of the medicine. It is not easy, and it is not in the blood of clinicians, but that's why I'm calling for early thinking and early consulting with regulators about designing of any even investigation-initiated small study. Thank you.
I just wanted to speak for Kara to just say that, you know, we would be extremely interested in discussions around using existing registries like the Kara registry to be able to use the patients that we already have and will have going forward, if that is going to be possible, as potential controls for studies, and whether that would be acceptable, you know, to the regulatory agencies, because currently we have over 600 patients with systemic JIA in the registry, and I believe that would be a very valuable resource to be able to use. So I would love to be involved in those discussions.